first, I would like to thank the management of Venkateswara University, the Vice Chancellor, the Principal, the Director, and Professor Sudarshanam in particular, and particularly to make all the efforts to group so many people and connect scientists, workers, and also hydroponic specialists, consultants around the world to make a wonderful interaction webinar. This may be the first, but we will continue further for further improvement of everybody. So when Dr. Sudarshan was asking me whether to do a presentation, I thought of giving some light on the mycorrhiza, arbuscular mycorrhiza, which has a predominant role and a lot of research is being happening on this aspect. In view of the controlled atmospheres in the hydroponic systems. Of late, the research in arbuscular mycorrhizal research is giving very, very promising results. So I thought I'll give you a light on, throw a light on this one aspect. So I'd like to do a small presentation. There are a number of slides, around 38 or 40 slides, but I'll run through fast and we can have an interactive session as well later. So I can save some time. Give me a time, I'll screen share my presentation, please. Yeah, 1 second. Okay. Yeah, it's okay. Yeah. Yeah, but this is sir, is that audible and also viewable?
Yes, sir. Go ahead.
Yeah. Okay. I'll continue then. Right. This is vesicular arbuscular mycorrhiza. Erstwhile, the name was like that. So many of the people are conversant on the name called VAM. Now it's called as arbuscular mycorrhiza. And also the mycorrhiza, the species level, a lot of changes have come. We now call them, most of them, as initially it was called as glomus, the genus, and now it's called rhizophagus. So I will go through the latest as a preliminary thing. The next seminars and all will go for advanced things, please. So it's called, this is the mycorrhiza is nothing but a fungus which colonizes near the roots, attached to the roots. Some of the species, they go inside the roots and colonizes in the xylem vessels, and they live there. And whatever there's an off late, the research indicate that there is a wonderful say what is a symbioncy. It's a microbial symbioncy between this fungus and the plant. Whatever the plant needs is communicated to the fungus, and fungus brings out. It goes to 10 feet down. 10 feet down. Many reports say that it goes up to 10 feet down. It pulls the micronutrients, and particularly the macronutrients like phosphate. Phosphate, if you have applied mycorrhiza, if your mycorrhiza is there, you don't have to apply the prescribed levels of phosphate. You can save 50% of the phosphate. So it gives an immense advantage for the fertilizer, economics of fertilizer. So this is the thing in the nutrients, and they have multiple advantages. Recently, in the last decade, a lot of research is being done in Europe. One is in Belgium. Belgium is one of the major centers that it have the mycorrhizal research for Europe. Erstwhile, it has started way back in US and Virginia, and we started the research in 1989, when we were helped by Virginian professors, Dr. Sylvia. She has helped us in giving the glomus species. IRA was also doing at the time, and we had a multi-locational and well-adapted strains given by the ATCC through Dr. Sylvia. We have tested around, even in the sugarcane. We have started in sugarcane and vegetables and a lot of commercial crops since then. And off late, Recent research is in IRI. IRI is going on in Philippines, and we came to know that a lot of advantages for the paddy as well. Even we are trying in paddy, and we have some two interesting photos from the farmer as well. I'll go through the slides, and by way I'll explain. Okay. Okay. So this is the way I was telling about the interaction between the mycorrhiza into the cortex vesicles and everything. It goes into this, and then my presentation is anyway available with the university. You can I will avail from there. I will run a little bit faster because a lot of slides are there. So there are two types of mycorrhiza, as I was telling, as ectomycorrhiza and endomycorrhiza. And most of the mycorrhiza now we are concentrating on endomycorrhiza. Ectomycorrhiza, they will perish and they will survive less due to biotic and abiotic stresses. And endomycorrhiza, they survive. They feed on phenolic components. Phenolic components are the excretory substances from the plant. Plants excrete phenolic substances, and that is the food of mycorrhiza. So it's not a parasite. It is actually they complement each other. Mycorrhiza and plants complement each other, and there's a wonderful phenomenon called glomus. Glomalin. Glomalin is a what you can a liquid secreted by mycorrhiza for exchanging of the vibrations, the messages. The messages are connected to the root through glomalin. Glomalin, so a lot of research is going on in glomalin. Maybe in the next maybe 5-6 years, some revelations will come out. A lot of research. And glomalin, we are trying to artificially enhance the the production of glomalin in the laboratory, in my laboratory as well. We are also doing some research on glomalin. So that is very important. If we understand the role of glomalin, then I think there will be a marginal or remarkable success in the application of mycorrhiza to all the systems. So to all the plants. So that's what the thing is, and I'll go through the next slide, please. Okay. So this is the difference I was telling, ectomycorrhiza and amycorrhiza. There is the classification is also different. Different classes are there in the ectomycorrhiza where they colonize, and endomycorrhiza, the particular classes only are going inside and colonizing inside. So this is the phases, different phases of infection of the mycorrhiza and development of the spores. The mycorrhiza generally has a hyphae, a stalk, hyphae, and also a spore, and that is actually is explicit here, sporulation. Let us go for the practical utility of mycorrhiza, not to the say the basics and biology of the fungi. These are called propagules. It is called the combined infectious propagules are combined with the hyphae, stalk, and the spore are called as propagules. And the government regulation is that so we need every gram of the mycorrhizal product, commercial product, should have at least say 100 spores, 100 spores, 100 spores, viable spores rather. That's right. So this is the actually Glomus mosa is one of the biggest spore they have, and this is actually the position. Very clear how it is attachment and everything. So this is one mechanism of action, how the infection happens, how it affects the root and everything. How it affects the root and everything. This is how a colonization happens. Then exploration, say how to touch the root. Certain plant species won't allow the mycorrhiza. Very few, like some of the timber species, there is no record of mycorrhizal establishment. But most of the forest trees survive only because of mycorrhiza, So this is the development, and these are all the photos that from different, we have collected samples from different areas, and also we have developed in our laboratory, micropropagated, and these are the photos from our laboratory. So there are different strategies of production of mycorrhiza. The one is growing outside. Erstwhile technology is growing outside in maize plants. Maize plants, they harbor this different genus and species of mycorrhizae easily. And we used to grow all the scientists started growing mycorrhizae outside with the maize. They used to apply the mycorrhizae to the maize, and maize colonizes, the roots colonize. So what they used to do after taking the crop, the root stock, only the bottom, the roots and the surrounding soil used to be collected, grinded, and sold as WAM infected area, or infected soil. So this is to be the first one, and later on, so it is applied to different crops outside, and then controlled atmosphere. So it's called as ex vitro. So in vitro, in the laboratory conditions, the latest technologies, the laboratory conditions, I'll go through and also show you that. And ex vitro is also there, like control, like hydroponics and all. It can be called as an ex vitro conditions. That's the greenhouse conditions and all. So you can see. So this is the ex vitro that you are envisaging, and they're also very successful. We can grow most of the graminae plants, family of graminae plants. They harbor and they have the symbioncy with WAM, ready symbioncy. So this is the flow chart that the cultivation of pot cultivation of spores. In the laboratory, is in vitro. In the laboratory, what we do, this is an example of carrot roots. So we develop the carrot roots, and this is called root hair culture. So what we do is, in particular medium, we grow the roots of carrot, and the technology, the basic technology lies in infection of the roots and growth. We to attain uncontrolled growth of the roots, hairy roots. Uncontrolled growth of hairy roots is a technology, and that technology was founded long back by Russians. Russians. There is a bacterium called Agrobacterium tumificans. The strain is called, I think, K34, K84. K84 is the strain, and with that strain, they produce nodules, root nodules, cancerous roots. So continuous development of the root, uncontrolled division of roots. We also need that type of roots, and we have our technology. Every microbiologist or biotechnology have their own techniques. Their technology, they infest. They see that the infection happens from by the WAM infection to this particular root. Then what happens is, as this root is continuous growing, it cannot stop the growth. The WAM mycorrhizal spores, which are attached internally to the xylem vessels and all, so they also grow. They take the phenolics secreted by the roots, and they live there, and they also grow. When they are applied as a product to outside, again when they come near the roots, they enter into that particular root of a crop that we are growing, commercial crop, and they also start the advantageous say work there. The symbioncy starts with that work. This is the technology, actually, basically, in brief. So this is one of the foreign laboratories, and I'll show my laboratory as well. So these are the photos of our laboratories. These are the photos of say the pigmentation. With the radiant technology, pigment technology, we have got the pigmentation where the spores and the reticulate nature of the mycorrhiza, the arbuscular mycorrhiza, were present in the root system. So electron microscopy photos. So this is our laboratory at Sujay Biotech. So we do the system and control laboratories. Everything is controlled there. So and we are happy to announce that the contamination levels is some days 0, and sometimes maximum is 1.5, 1.3, something like this. Very marginal. The government rule is less than 5%. We are almost less than 1% at any point of time. This is our laboratory. So where we grow uncontrolled roots. You can't see any shoot system. Only root grows continuously. This is the specific media for specific genus of the fungus, and it grows. The growth period is 90 days. After 90 days, we can harvest the spores there, and there is a methodology of harvesting and say mechanical shakers, and we'll use some buffers to dilute the medium. and also second, it's also every batch, every randomly is checked for the sporulation. Number of spores, enumeration of spores is done by this one, and we get a graph also how much is the strength of. We can the advantage of VAM is we can sell the VAM, we can concentrate the spores and sell. So for 1,000 acres, we can give in 1 kg. 1 kg root biomass of VAM will contain, say, for 1,000 acres, sufficient VAM spores for 1,000 acres. So the logistics will be, saved. A lot of logistics will be saved. It can be further diluted with any, say, soil media and then applied to the crops. And this is the way simple methodology of in the laminar flow. Well, it's after during the harvesting here. It's a harvest. So this is the media that is diluted, and some of the spores present still there in the root system. A lot of spores will be the root system. A lot of spores will be precipitating in the medium, in the specific medium. So both are separated and both are filtered, and the spores are separated. All the mechanical things. So and then dried up a particular temperature, and this is the root biomass. Root biomass of this one has full of spores. Almost 125,000 spores per gram. So this sort of things, So every batch we get about 125 grams. And 1 kg, I was telling you, will be catering it to 1,000 acres. So this one. So this is the powder from this packet. When it's tested, it looks like this. You can see number of spores. It's called propagules. This is again ex vitro, that is applied to the ex vitro to grow as a VAM. This is one of the procedure, and the second procedure is in vitro. So this is what I was talking about. This is a farmer's photo. One of our farmer was using it for paddy. So paddy, the structure of the root has changed. The graminary roots are generally fibrous, and because it's an advantageous root system, it's a fibrous than in paddy or rhizocytic. And what we have seen is most of the roots are becoming very thick, stalk-like, very, very astounding. And he was amazed, what is happening here? Is there anything problem with this? And he has complained to us. He said, there's no complaint, you'll get more crop. He got 450 kg, 6 bags extra per acre. He has 10 acres, something like that. He was much benefited. He was very, very happy. Ever since word of mouth, we are getting good orders for paddy as well. So this one, the right side one, you can see on the black color and see number of roots. What the farmer has done, the another farmer, he has purchased VAM from us in bulk. In a gunny bag, he has put it in gunny bag, mixed with some, say, poultry manure or something. He put it in the bag. So you want to put it in the, say, at the root zone of the plants, banana farmer. So, but what happened is he forgot it. It was a rainy season, forgot it. He is kept at the bottom of a eucalyptus in his farm. So that is the VAM has seeped inside the soil, and after about say about 1 month, he has taken out the bag. He has recognized his bag. No, he forgot, and he has taken out the bag, and he has seen the roots of eucalyptus are spread outside. This is outside the on the soil, below the bag. This is not the spreading. This is a real, actually a wonder. So we can see the initiation, the work, and what the root development. We have seen practically by the farmer at the farmer level. This is one of the wonderful photos. We have shared it to almost all the renowned scientists in Taiwan, in Belgium, in US, and all. They are also very, very happy because the school of thought of VAM. So they have worked for more than 40 years, very, very hard to see these sort of results. More astounding results are coming. Fully advantageous. You spend a little, you got to get a maximum profit. See, coming to role of VAM in hydroponics. Actually, this is the actual topic that I was promising Dr. Sudarshanam Garu, Professor Sudarshanam. So actually, this has been tried out in different areas, different places, different places around the world. Particularly in Southeast Asia, is very, very successful. And Europe also of late is coming with a lot of hydroponic systems in the this one. And then, So basically, we have to discuss the most important thing. What from our side, we want to see a very important thing here. Discuss one thing here. This is the level of oxygen, the percentage of oxygen present in the hydroponic system is directly proportional to the growth and performance of the mycorrhiza and also performance of a plant. So I have seen in Israel and see in Israel the hydroponic system. They use a lot of oxygen, artificial oxygen to provide. They're providing a lot of oxygen, not a simple blower, not an air pump, because air contains only 16% of oxygen, and out of which in polluted towns and cities we may get a 14, 13, even 12, 10, 11 even in the industrial areas. But they are providing pure oxygen. Pure oxygen. They see that at any point of time, the DO levels, the dissolved oxygen levels present in the water should not decrease than 7 ppm. This is what I was told in Israel. So here we are doing a lot of experiments. We're seeing 7 DO, 8.5 DO, and 4.5 DO, and 2 DO also. And we see the performance of the roots, interaction with the mycorrhiza, and also absorption of nutrients. We analyze this one. That experiments are not yet concluded, still going on, and we see a lot of say advantage of presence of oxygen. And we will also come out with a research paper, and we will try to publish, maybe with the help of Professor Sudashanam, because it's a collaborative thing. So we'll do this. Very good advantage of oxygen. Yeah, this is all what I was talking about, oxygenation of foods. So commercial hydroponics, a lot of firms are there. See a lot of countries like Netherlands, Spain, Canada, USA, Mexico, of course, Japan, Australia in particular. They are very strong in glomus research, resistant glomus. That's a wonderful revolution. They do on eucalyptus, they're doing on soybean, they're doing on, in particular, some of the oil producing crops. They're very strong. So on each country, if they spend some time, energy, and money on research in mycorrhiza, I think it will be much more advantageous to the further generations and the farmers in particular. Even in the Middle East, a lot of people see to withhold the desiccation of the plants due to lack of water and also presence of hard waters, because most of the water in the Middle East is recycled water, and municipal sewage is recycled after the RO treatment, and because that water becomes a little bit hard, the chlorides can neither be settled nor be removed easily. So that will be there. So hard waters pose a problem to agriculture. That is known to everybody. So this is why when the WAM is there, that problem also is decreasing. And also the latest research in Indonesia and also in Taiwan, what they say, plant protection mechanism is totally governed by certain traits and secondary metabolites produced by mycorrhiza, the fungal mycorrhiza. So that's a great revolution. The governance of the plant protection is supported by mycorrhiza. When mycorrhiza is there, the plant protects itself very well, and when mycorrhiza is not there, it succumbs. So we still don't know the real mechanism and real contribution of WAM. And they were trying to, in Australia, they're trying to, record the messages. Long back, or Jagdish Chandra Bose has recorded certain messages from the plant to plant. Here, we are trying to understand the messages from the plant to the fungus. The plant, if it needs zinc, it gives an indication to mycorrhiza, and mycorrhiza goes down, and it pulls up, assimilates the particular zinc and supplies to the plant. There are a lot of messages are going on. That recordings are going on. A lot of basic research is still continuing, that to the advantage of the humans everywhere. The Bengal system of hydroponics. This is called paddy cum fish culture, was in practice since 1961. 61, long ago. The paddy fields where they used a fish called chanos, milkfish, chanos. Indonesia is also very famous for this paddy cum fish culture. There, when mycorrhiza is applied along in the paddy, the excreta given by dropped by the fish also also being a major advantage to the growth of mycorrhiza. The growth is exemplary. Exemplary. Practically, it's impossible. It's proved impossible to get the same amount of spores in the laboratory. That's a real wonder. So that's why our scientists have prepared this one a small note. This is regarding the Bengal type actually. Gujarat also adopted this technology, and exotic hybrids of roses and all, and they're 100% exporting. So everybody is using my mycorrhiza. When they're using mycorrhiza, the root growth is immense, very luxurious, and the absorption and assimilation of nutrients is very fast. So the conservation of nutrients, plant protection, conservation of say phosphate, and also micro pulling up of nutrients. So when I go, earlier crop they use fertilizers, more fertilizers. Some are seeped down, percolated down. All can be again pulled up. So it's a major advantage. These are the crops. Of course, my earlier speaker, Prasad, they have done very good work. They were, all were tried, I think so. Yeah, leafy vegetables, certain medicinal crops. I'm also interested to see my next speaker, the last speaker. I think so. In this, and I've seen somebody's presenting on the medicinal crops. That is also has major advantage. A lot of research is being done on medicinal crops in this aspect, even in Malaysia. Yes, ornamentals, everything. These are the certain photographs my scientists have collected from internet. It's not mine. So all the internet photos, co-researchers. So it's an exposure to everybody in this particular aspect, because this is a small aspect that is coming up. Yeah, the infection I was telling about, the infection is being controlled, like infections of Fusarium, Pythium, and Phytophthora. It's very common in Solanaceae families. Tomatoes succumb to this very easily. So that can be controlled. When the mycorrhiza is there, there'll be no infection. The infection rate is very slow, and then we can control. We can identify and control. So most of the crops we have seen, when VAM is there, the survival is much better, and thriving plants thrive much. Even in bananas, in horticultural crops, we have seen the presence of VAM is making a lot of difference. These are the cucumbers. Cucumbers were one of my say fellow scientists. Also, they're trying in Zahirabad and all this area. And they say it's very good. It's controlling a lot of pests there. Yeah, I think the same thing is repetition. The silicon-based plant defense mechanism. There is one type of other research called that plant defense mechanism is governed by mycorrhiza. We see activation of silica present in the soil or applicative in the liquid also. That is one of the reasons. So attacking the pathogen. The carotenoid. This is one of the this is one of the reason that we have seen. So one second. I just want to explain. Yeah, this one. This is the soil-based color development. We have seen a wonderful say research advantage on this with mycorrhiza. When mycorrhiza is applied for the cut flowers and for the vegetables, the impact of the color has increased. The annotation is improved a lot. So cut flowers has a major advantage due to this. So this is also a lot of research is supporting into this. Even in Latin America, Brazil, Embrapa is doing a lot of research on this. Even Chinese people have done a lot of work, but their publication, most of the publications are in Chinese and Mandarin. So it is a little bit difficult to get communicating with them. So only Shandong University is coming out, but most of the universities they don't say interact. That's the issue there. So this is a major advantage. This is one more advantage rather. So this is like cut flowers. A lot of experiments are done on this, the coloration. This is onions also major advantage. We can see the differences of onion and also chilies. Solanaceae family give a major say infection. Even ginger also we have seen a lot of ginger, and of course this is one of the very common things. Strawberries, they respond to VAM very easily. Wheat actually haven't seen a lot of people are doing in Punjab Agricultural University, they are doing, but wheat grass is showing a lot of infection. It's growing very, very fast. Wheat grass like this is used as say for fodder crops. Even in maize seed also is used as fodder crops. Whatever the seed maize collected for the seed production is generally packed, and which are rejected seed for as a seed to the next generation are grown donated to the people for the fodder crops. All the arid regions in our state also, in our state of Andhra Pradesh, in India, major arid regions, fodder crops can be developed, and for the summer, the cattle can be fed. This should the government initiative is needed. We have earlier we have discussed it with this, Dr. Sudarshanam. So put it. I have in myself. We have put a paper to state government. We'll stay waiting for that nod. Yeah, a lot of literature is there. This is regarding a lot of literature. You can pass through later. So thank you very much for giving a very wonderful opportunity. If anyone needs any assistance or any reference of my research, definitely we can contact through Dr. Sudarshanam. We are contacted. All my address and everything, all contacts are with the Professor Sudarshanam because we are collaborators. So if there are any queries and everything, you can write in Urban Kisaan, all my this one urban farming. My details are also there. My mails and everything are there. Any specific thing is there, you can contact Dr. Sudarshanam as well. We are doing collaborative research, and I thank once again the management of Venkateswara University, principal, director, and in particular Professor Sudarshanam for giving me a wonderful opportunity to present a small technology that we are developing and research to the welfare of the farmers. Thank you very much.